Our second speaker is John Hartwig, and the title of his talk, and again, obviously, John doesn't need any introduction. He's a professor at University of California, Berkeley, and the title of his talk is Catalytic Functionalization of CH bonds with Main Group Reagents. All yours, John.
Okay, can everybody hear me okay? Audio okay?
It's fine, John. It's fine.
All right. Okay, well, thanks, Hugh. It's a great idea to hold these events, and it's a real pleasure and an honor to participate, particularly with today's group of speakers. So today I'm going to talk about CH bond functionalization in my group, obviously. I guess I intended when I gave you a title many months ago to focus on the reactions of boron and silicon reagents, and I will talk about that in the beginning of my talk, but I'll also talk about some recent results on the azidation of CH bonds as well. So if I can have my first slide. Second slide. So it's a short presentation, so I don't have a long time for introduction, but I did want to talk a little bit about the strategy that's evolved from our group in the functionalization of CH bonds that's depicted on this slide. And that is to develop reactions where we install what we've called a temporary functional group, or some functional group that can be converted into products containing new carbon-carbon bonds of various sorts, carbon-halogen bonds, carbon-nitrogen bonds, and carbon-oxygen bonds. Next slide, or next animation. And so the bulk of our work in this area over the years has been done on the development of catalysts and conditions for the functionalization of CH bonds to make carbon-boron bonds and aryl and alkyl boronate esters. And some of the most practical systems that are based on iridium catalysts with nitrogen ligands, ditert-butylbipyridine, tetramethylphenanthroline ligands we've worked with recently to make aryl-boron derivatives that can be converted to a variety of products. Next. But the development of this chemistry has also inspired us to think about other kinds of CH functionalization reactions that could be used in this sort of a way. And so Patrick Fear and my group developed a reaction, silver-mediated reaction of silver 2-fluoride to generate 2-fluoropyridines with the original intent really of incorporation of fluorine into aromatic systems for medicinal chemistry and the value of the aryl fluorides or heteroaromatic fluorides themselves. But we came to appreciate, of course, that the carbon-fluorine aromatic or heteroaromatic carbon-fluorine bond is the most active for nucleophilic aromatic substitution chemistry. And so what we have in the second paper listed here is the combination of this fluorination and then substitution to make a wide variety of products from the second step of that transformation. Next slide. But what I want to spend the bulk of the time today talking about is the functionalization of alkyl CH bonds. And this does have a relatively long history in the group, right? Some of the very first chemistry we did with metal boreal compounds involved the functionalization of the primary CH bonds of alkanes under photochemical conditions to generate alkyl boronate esters, in which the only product deserved is the product from functionalization of the primary CH bond. This is a stoichiometric reaction. It's photochemical, but that did provide a platform for the development of a catalytic process a few years later to functionalize the primary CH bonds of alkanes with boron reagents in the presence of a rhodium catalyst, which maintained this CP star metal fragment, but now with a labile hexamethylbenzene ligand. So the real question at that time then was, how could we develop this functionalization of alkanes into something that would be more synthetically valuable? Because both of these reactions were first published just with alkanes and with neat alkanes, an alkane solvent. And so pretty far away from something that a reaction one would think would be synthetically valuable, at least for medicinal chemistry type purposes. Next slide. And so more recently, we've been working on the functionalization of CH bonds with Silanes, and Eric Simmons, a postdoc from the group, developed a scenario in which he would use the existing functional groups in a molecule as a sort of anchor point for the reagent to functionalize a CH bond. So next slide, or next animation. Right, and so the concept was to, say, take an alcohol or a ketone, two of the most common types of molecules in organic chemistry, of course, and then to silate the oxygens by either dehydrogenated silylation or hydrosilylation. And now it's poised for an intermolecular silylation of the CH bond with the iridium catalyst that we've been using for the borylation. And the product may look like a little bit of an exotic structure, but because it has a silicon-oxygen bond, it can undergo oxidation here to generate a diol product under Mal-Fleming type of conditions. And so if we think about trying to do that with boron reagents, I don't know how we would do that, because the kind of intermediate we need to make would have a boron-oxygen bond, a hydride, and some sort of substituent on it. And of course, those kinds of molecules are unstable to disproportionation. So for the functionalization of alkyl CH bonds at the primary position, these kinds of molecules, we focused on using silicon reagents. And I don't have time to go into the details of this chemistry. Next slide. But I did decide to pull out a few examples that demonstrate some of the value of this for site and stereoselective functionalization. So for example, Eric showed that with fenchol, the reaction would occur diastereoselectively at one of those two geminal methyl groups, and to generate this single isomer in about 60% yield. And similarly for camphor, you can hydrosylate the ketone, and then that catalyst will then reach around and grab the hydrogens on carbon 10 to generate the diol, and then protection and isolation gave a single isomer of the product. And then as we began to look at more and more complex substrates, methylolenate shown here has, of course, many types of CH bonds, primary, secondary, and tertiary CH bonds, but it also has functionality that could be reduced by silanes and catalysts, like an alkene and an ester. But with the iridium catalyst, and in some cases ruthenium catalyst, we've been able to silate the oxygen of the alcohol, right, to make a silyl ether, and then there's a diastereoselective reaction to functionalize one of the two methyl groups, and then oxidation to generate the diol product. And so this is a sequence that had been done in many steps previously, but just with this one pot sequence, one's able to then generate products, which of course have very different solubilities and polarities than the starting materials. Next slide. So recently we've been looking to expand the scope of this silylation chemistry as well as other types of alkane functionalization. And so B.G. Lee, in a collaboration with Matthias Dries during a time of my Einstein Fellowship in Berlin, began to work on the silylation of secondary CH bonds. And so the general scheme is shown at the top of the slide, where one would have a secondary or tertiary alcohol, silylate the alcohol, and then in this case, with a longer chain, reach around and grab a secondary CH bond instead of primary CH bond to make the carbon-silicon bond. Next slide. And what you can see here is that if you just have a relatively unconstrained secondary alcohol, silylate that the yield is quite low for silylation of a secondary CH bond, in contrast to what we see for silylation of primary CH bonds. But if it's a tertiary alcohol, then there starts to be substantial yield of the product, low DR, but some measurable yield. And of course, next slide, this is just a manifestation really of the Thorpe-Ingold effect. This is a cyclization reaction after all, and so having some gem disubstitution will help that. But what BG found was that if you simply increase the size of one of the substituents a little bit more, and we go from a pretty modest yield to essentially quantitative yield for silylation of the secondary CH bond to make this 5-membered ring silicon cycle, which then can be subject to oxidation to make diol products again. So next slide. We have also been able to develop conditions for the silylation of the secondary CH bonds and secondary alcohols. Now again, if it's just an unconstrained secondary alcohol like that for heptanol, then there's very low yield. But if you have a secondary alcohol that admittedly is a bit of an engineered substrate, but does have a conformational restriction to it, then the reaction can occur in good to excellent yield with very high diastereoselectivity. So certainly the challenge now is to develop a catalyst that's a bit more active for this transformation so that we can begin to see the kind of functional group compatibility that we saw for the silylation of primary CH bonds. And that's something I think we can accomplish because the reactivity of secondary versus primary is about 50 to 1. So not by any means an insurmountable difference in reactivity of the two. Next slide. So that's just sort of a quick way to set the stage for what I'd like to talk about next. And so what this shows is that we can functionalize primary CH bonds in an undirected way with the borylation chemistry, and we're working on, development of functional group compatibility and so forth for that. We can do silylation of primary CH bonds directed by an alcohol, now secondary CH bonds as well. And then you can convert those products to a variety of different final products containing a hydroxyl group, an amino substituent, an aryl group through cross-coupling. But of course, for the silylation chemistry, we are a bit limited in that one can not convert a carbon-silicon bond to a carbon-nitrogen bond of an alkylamine yet, at least, under any conditions. So, given my group's interest in amination chemistry, we have wondered whether we could develop chemistry to do amination of alkyl CH bonds, and also to be able to affect transformations at a tertiary CH bond, given that we've now developed chemistry of primary and secondary CH bonds. Next slide. And so the question then is whether we can develop a reaction that would install a nitrogen-based functional group, but really to do that in a way that would be a nitrogen-based functional group that could be converted to a diverse array of products. Now, of course, we're by no means the first group to think about the amination of CH bonds, and in your own NSF center, you got Justin Dubois, who's been a leader in this area, along with Dodd and Dalban, who've developed a lot of amination chemistry over the years, and these are just two references to review articles by those groups. Next slide. And so Ankit Sharma, my group, who's sitting on my left over here, was thinking about, what kind of functional group to incorporate that would have this kind of, at least begin some sense of the diversity of what we could do with silicon and boron. And so we thought about the azidation of CH bonds, where the CH bond in an alkyl substrate could be converted to that carbon-nitrogen bond, and then the product could be used for other CH bond functionalizations, or reduced to amines and amides, or you undergo cyclization, or used in heterocycle synthesis. Now, next slide. So to some extent, an azide can also be useful itself in medicinal chemistry, of course, AZT being the most famous azide molecule in that arena. Next slide. So again, many people have thought about this transformation, or at least some people have thought about this transformation over the years. Over 30 years ago, Craig Hill had published on using iodosobenzene with an azide source, that combination with metal catalysts for doing azidation, and as did Larry Kay and Miguel Costas over the years, and Jay Groves in your own center very recently publishing in JAX on this. But for the most part, this chemistry has been done with a combination of azide and then iodosobenzene, giving a mixture of the alkyl azide and the alcohol product. Next slide. Okay. And so Ankit then began to think about whether there was a way to combine the oxidant and the azide source in the same reagent, so you wouldn't have this competing process to generate alcohol along with the azidation chemistry. Next slide. And so Ankit was aware of the chemistry published by Zedankin in 1996 on this hypervalent iodine reagent containing the azide that would fit that criteria of having the oxidant and the azide together. The reagent is stable up to about 130 degrees, and so for operating under mild conditions, it should be safe. We haven't done a lot of tests on shock sensitivity and so forth, but there's an exotherm at 130, but as you'll see, we're operating at much lower temperatures than that. Next slide. And so the other thing that Zedankin had shown is that that reagent does have the thermodynamic potential to functionalize an alkane. So in the presence of a radical initiator, cis-decalin is converted to its corresponding azide from functionalization at the tertiary CH bond. And so the question then is whether we could not use just a radical initiator, but could we develop a catalyst that would make that reaction occur under lower temperatures than refluxing dichloroethane, and with selectivity or stereoselectivity that could be controlled by the properties and architecture of a catalyst. Next slide. So we began to look at potential catalysts, and if you go back and look at the porphyrin systems that would give a product from the combination of oxidant and azide, they were pretty poor, very low yields for the reaction with this hypervalent iodine reagent containing the azide in it. And so it began to then, next slide, look at metal salts, simple metal salts, and began to at least see some measurable amount of products with various iron salts. And so of course the way to modulate the reactivity is to put a ligand on that metal other than just the X group. Next slide. And so looking at a variety of nitrogen-based ligands, because this is oxidation chemistry, so based on ligands based on nitrogen, what Anki found was that you could get good yield for the azidation of this decalin with this tridente pibox type ligand giving 75% yield. So that was our starting point for looking at the scope of the azidation chemistry. Next slide. And so it's been well established over the years that for electrophilic species undergoing oxidative chemistry, that there is a electronic preference for one type of CH bond over another and relative to its position to functional groups, usually electron withdrawing functional groups. And so that's, you know, as many of you know, been exploited by Christina White with iron catalysts recently, and also been reviewed by Baron and Newhouse and with a variety of different reagents and the effect on that. So, Anki began to look at whether this azidation chemistry would be subject to the same kind of trends in terms of the selectivity for a remote or proximal tertiary CH bond to electron withdrawing functional groups. Next slide. And so this also shows regioselectivity, but also shows the functional group compatibility of this chemistry. And so the reaction occurs in the presence of, you know, esters and carboxylic acids and amides and halides and nitriles and ketones. And what you can see is that there's actually quite substantial selectivity for the distal CH bond over the proximal CH bond to these electron withdrawing substituents on the alkyl chain. So with that background, right, we began to look at increasingly complex molecules. Yeah, next slide. And the selective, both the site selectivity and again, functional group compatibility. So you can see the reaction tolerates epoxides, azides, aziridines, cyclopropanes, and the reaction occurs at the tertiary CH bond that's distal to these electron withdrawing functional groups. It also occurs at benzylic CH bonds. You can see in the bottom left that a secondary and tertiary benzylic CH bonds. And this estrone derivative, you'll see this again later, at a benzylic CH bond in that, as well as in podocarpic acid, this derivative. Next slide. So recently, Anki has been working with Rashad Karimov, who's on my right here, at the modification of sort of increasingly complex molecules and looking at the functionalization of a variety of natural products, and in part to look at the scope of the chemistry, but also to try to begin to get some guidelines on understanding what the site selectivity will be. And so you'll see on these slides the sort of emerging guidelines here showing that benzylic CH bonds react preferentially over alkyl CH bonds and the selectivity for different types of benzylic CH bonds. And so that's shown by the substrates here, where with these estrone derivatives, you can see reaction at benzylic CH bonds. Although for reasons we don't fully understand, the major product from the TBS protected example is at the benzylic CH bond shown in blue at where the azide is. But for the difluoromethyl substituted case, it goes after the tertiary CH bond instead of the secondary benzylic CH bond. So we don't quite understand that, but you can see that the identity of the substituents can affect the site selectivity substantially. And then for the tocopherol example on the bottom, you can see selectivity for secondary versus primary benzylic CH bonds. You know, there's a small number of primary, just 3 versus the 9 primary benzylic, or 2 versus the 9 primary benzylic CH bonds, and still giving us a major product, the one from reaction at the secondary position and with perfect selectivity over reaction at the non-benzylic tertiary CH bonds. Next slide. And this slide then shows examples of reactions at alkyl, not benzylic, but at tertiary alkyl CH bonds. And again, the reaction occurs away from the electron withdrawing functionality. So in this cycloheximide, we get a decent yield of product from reaction at the tertiary CH bond that's away from the imide and ketone functionality. And digoxygenin, the reaction doesn't occur at the allylic position. I'll touch on this again later because that's withdrawing. It goes at the tertiary alkyl CH bond. And you can sort of peruse through examples. There's several products with artemisinic acid derivative shown at the top right, but highest reactivity away from the embedded ester of the lactone. And I guess for betulin, the reaction, you might expect it to occur at the exocyclic tertiary CH bond, but actually if you think about the conformation of that molecule, that substituent in the CH bond lies underneath the molecule, so it's actually, we think that the reactivity is occurring at the most sterically accessible of those tertiary CH bonds and away from the withdrawing acetate functionality. And of course, as you can see, comparing the acetate protected alcohol as well as the ester, the identity of those substituents can have an influence on the relative reactivities. And so finally, you might imagine that molecules with an alkene in them that have allylic CH bonds would also react because allylic CH bonds are typically weak, and that is the case. And so in matacasic acid, the reaction occurs at the allylic CH bond, the secondary allylic CH bond. And pinene also a reaction at the secondary or primary allylic CH bond, although modest yield. But the example I showed you earlier of digoxygenin, the reaction occurs at the tertiary alkyl versus the allylic CH bond because that allylic CH bond is next to an alkene that's an electron poor, electron withdrawing alkene. And in case this gibberellic acid example, in the case of a 1,1 disubstituted alkene under these conditions, we see diazidation. And so that's certainly a transformation that we're interested in developing, effectively a diamination of an alkene. Right, so some emerging guidelines here then. I guess we'll move forward since we lost it. That's okay. Emerging guidelines here. So tri-substituted olefins undergo allylic azidation. The 1,1 disubstituted olefins undergo diazidation as opposed to allylic and azidation, and the steric hindrance and electron deficient olefins are unreactive, as shown by the case at the bottom left. Next slide. So I won't go into detail on this because, of course, people know what azides can be used for, right? They can be converted to a variety of different products, but one can attach, say, fluorophores to track a molecule or recognition elements for creating affinity columns, as well as using that to incorporate polar functionality, such as in positions of molecules where there's mostly hydrocarbon. Next slide. So I just want to finish by touching on a couple of mechanistic issues. We're just beginning some of our mechanistic studies, but I can tell you a little bit about what we know so far. So the proposed cycle would be that you start with an iron 2 compound that reacts with the azide to generate the iron 3 azide and a radical. And then that organic radical would extract the CH bond to generate an alkyl radical. And in the absence of a catalyst, so if this radical were generated in the absence of the iron, then that alkyl radical would just react with the azide reagent and as in the original chemistry by Zidonkin. But the concept, at least, was that in the presence of the iron compound, that alkyl radical could be combining with the iron azide to generate the alkyl azide and regenerate the iron catalyst. So some evidence is that we have done the reaction of the iron 2 plus the azide reagent, and that does generate an iron 3 azide that's been characterized. So the question then is, what happens to that alkyl radical? Does it really react with the iron azide that we had isolated? Next slide. So a couple pieces of evidence in case you need convincing that the reaction occurs to an alkyl radical. If you start with cis or trans decalin, you get the same diastereomeric mixture of products out 6.3 to 1 under these conditions. If you start with cis and trans mixed, cis or trans dimethyl cyclohexane, you get the same DR for the product of that reaction as well. And if you start with a pinane, which is a 3 to 1 mixture of diastereomers, epimeric at that CH bond that's reacting, you get a 50 to 1 ratio, roughly 50 to 1 ratio of diastereomeric products. So that's actually perhaps synthetically valuable as well as mechanistically revealing. Next slide. So then what happens with that alkyl radical? Right, so we have some preliminary evidence to suggest that we can control stereoselectivity for the azidation chemistry. And so if you look at the two examples on this slide, the two reactions are done with the opposite enantiomers of the ligand on the iron. And so the reaction to form this diastereomeric mixture of products at the top is done with the SS version of that pi box ligand, and that gives a 6 to 1 ratio of the diastereomers favoring the one shown. But if you use the opposite enantiomer of the ligand on the iron, then you get a 3 to 1 ratio of diastereomers favoring the opposite epimer at that position. And so maybe this in the end will be synthetically valuable. We hope that this will be kind of a general trend, that we can control diastereoselectivity selectivity through the absolute configuration of the ligand to the catalyst, but mechanistically, at least it shows that the CN bond is being formed within the coordination sphere of the iron ligand complex. Next slide. So that's what I wanted to tell you about today. And just to sort of summarize conceptually, right, we have some chemistry with main group boron and silicon reagents that can occur at primary CH bonds. We're beginning to develop that chemistry at secondary CH bonds, but have now developed some chemistry that occurs at tertiary CH bonds, installing an azide group that can be converted to a variety of different products containing that carbon-nitrogen bond that's been formed by this chemistry. Next slide. So what remains is to thank the people involved in this. So I've highlighted Karen and Huyan, who always, were the very initial people to work on the borylation, stoichiometric, and catalytic chemistry, getting us started in this general area. And I talked about Eric Simmons' work on the Silylation chemistry, and then Ankit and Rashad on the Azidation chemistry for the second half of the lecture. So thanks very much to them and their contributions and conceptually and in the lab. And thanks to the NSF and NIH for support of different aspects of this chemistry over the years. And of course, thanks for your attention and for the invitation.
Thank you, John. That was very interesting indeed. So we have a question from Stanford. And the question is, from what you've seen so far, is this azidation chemistry compatible to densely oxygenated substrates like protected sugars and deoxy sugars?
I guess I could say that we're working on that. And there are some weak CH bonds in those substrates that would be alpha to oxygen, which are kind of an issue. So some sugars work okay, some don't. So I didn't present on that because it's not all ironed out yet. But I think there's definitely hope. But I don't know how general that will be. But we got to develop some guidelines and some rules before we talk in much detail about that. But it is possible.
Okay, a question from me. So it's really impressive the type of selectivity you can have in really pretty elaborate substrates. And it looked to me as if your yield was of the clean material, but is there a challenge, a separation challenge associated with these compounds? How do you go about getting clean material from them?
Well, I guess there's a couple of challenges. One is conversion, right? So we don't always have full conversion. But part of the reason for that is that if you push the reaction to full conversion, you can get reaction at multiple sites, right? So the molecules have many CH bonds in them. And so then the question is, you're better off trying to separate, have a mixture of some leftover starting material and monofunctionalized, or do you want to have unfunctionalized mono and difunctionalized and separate those? So there's definitely a bit of adjusting of conditions to get a product, solution that allows for separation. So I guess I'm not in the lab anymore, so I don't know, maybe Ankit can comment on and Rashad on separation issues. I mean, we're just using columns, but I don't know how difficult to select this separation is. I don't think it's that difficult to separation. I think it's really an issue of trying to adjust the conversion so you don't have multiple functionalization products from different positions. Right.
Okay, a question from Worcester Polytechnic Institute. Can you sequentially use the borylation silylation chemistry in combination with the azidation chemistry?
We have done borylation and azidation.
First borylation, then azidation. Or
we have used borylation substrate for azidation, and that works fine. So pinnacle boron is tolerated.
Oh, I see. Yeah. So Ankit's feeding me some information here. So the azidation chemistry will tolerate a pinnacle boronate group on the substrate.
So one could imagine.
So we haven't done it, but you could imagine doing a borylation to install the B-pin fragment on a molecule, then doing the azidation chemistry, and then manipulating those two groups independently.
And Chen has shown that
in the
borylation and silylation.
What's that? Yeah, I mean, you can certainly do the borylation and then silylation. The silylation will tolerate the B-pin reagents also.
Which is iron cap
disappeared. I missed that, Hugh. You cut out for a second.
Okay, I think it froze for a second. Yeah, the question is from Kais. Is ion catalyst so special for, why is the ion catalyst so special for the azidation? Meaning that can other catalysts work as well from mechanistically?
Yeah, I guess we don't know the answer to that question. I mean, I would never say that a catalyst is unique. Right? Over the years, you always think you've got the best system, and then you find something else, or someone else finds something else. So we have, as the mechanism stands, right? There's one electron redox. You generate a metal azide, and the alkyl radical combines with the metal azide. So I would say by no means does iron seem unique for that, or iron with a pi box ligand on it. That's a bit empirical why that ligand works. So we don't have enough mechanistic detail to really, and understanding of speciation of the different compounds in there to know at this point, or predict at this point, what would work and what wouldn't work. But I find it hard to believe that it would just be this iron catalyst in the end.
Okay. Well, John, thanks very much. A wonderful talk. Well done.